Good evening, everyone. It's a great pleasure to present this work entitled From Environmental Remediation to Energy Storage, the evolving role of bio-based composites. So in this presentation, I will share research journey based on the development of bio-based composite materials for different sustainability-related applications. So the main idea is to show how biopolymers such as alginate-chitosan and related bio-based matrices can be engineered into multifunctional materials. So these materials were first explored for water treatment, then extended to smart agriculture through slow release fertilizer systems, and more recently redirected toward energy storage applications. So this talk is organized around one central question: how can the same bio-based platform be tuned to address different environmental, agriculture, and energy challenges? So this slide summarizes the overall logic of energy of our research journey. So the first step is water treatment. In this part, biopolymer composite bits were designed as eco-friendly adsorbents for removal the pollutants such as heavy metals and organic dyes. So the second step is smart agriculture. Here, the same material concept was adapted to control the release of nutrients and improve fertilizer efficiency. So the third direction is energy storage, where bio-derived carbon-based composites are being explored for clean energy technologies, especially supercapacitor electrodes. So the important message is that these three applications are not disconnected. They are linked by the same design philosophy. One tunable biopolymer-based platform adapted from pollutant capture to nutrient delivery and finally to energy storage. So before presenting the applications, it's important to explain why biopolymers are suitable for this work. So biopolymers are attractive because they are renewable, biodegradable, and generally less toxic than conventional synthetic polymers. More importantly, they contain reactive functional groups such as carboxyl, hydroxyl, and amino groups. These groups are essential because they can interact with ions, pollutants, nutrients, or active phases through adsorption, complexation, gelation, or ion exchange. Another key advantage is their tunable architecture. By controlling cross-linking, grafting, or compositing, we can adjust porosity, diffusion pathways, and ion transport. This means that biopolymers are not only natural materials, they can be engineered as functional platforms for environmental remediation, controlled nutrient release, and energy storage. So our first application focused on water treatment using alginate-chitosan hydroxyapatite hybrid beads. So these beads were prepared by ionotropic gelation in calcium chloride using hydroxyapatite derived from Moroccan natural rock phosphate as a local mineral resource. So the formation of the hybrid structure was confirmed by characterization. FTIR analysis highlighted the presence of the main functional groups from alginate, chitosan, and hydroxyapatite, while XRD confirmed the crystalline contribution of hydroxyapatite within the biopolymer matrix. So after adsorption, SIM observations also showed visible changes on the bead's surface, supporting the interaction between the pollutants and the active sites of the composite. This synergy between carboxyl groups from alginate, amino groups from chitosan, and ion exchange sites from hydroxyapatite explains the high adsorption performance, reaching about 400 454 milligram per gram for methylene blue and 208 milligram per gram for copper ions. So the beads also showed good regeneration ability with a stable performance over 5 desorption cycles and around 80% efficiency retained. Overall, this study validates our bio-based composite beads platform as an efficient, low-cost, and eco-friendly adsorbent for wastewater treatment. After validating the biopolymer bead platform for water treatment, the next step was to explore whether the same material architecture could be transferred to another sustainability challenges, smart fertilization. So in water treatment, the bead network controls pollutant diffusion and adsorption through porosity and functional groups such as carboxyl, hydroxyl, and amino groups. The same principles can be used in agriculture, but with a different function. Instead of capturing pollutants, the material is designed to retain and gradually release nutrients. So the transition is based on a simple scientific bridge from pollutant adsorption to NPK controlled release. This means that the same biopolymer platform can be re-engineered by tuning the matrix structure, ion interactions, and diffusion pathways in order to improve fertilizer efficiency and reduce nutrient losses. So this second application focused on the development of bio-based coating for fertilizer, coatings for development of bio-based coatings for slow release fertilizers. So here the objective was to use chitosan polyvinyl alcohol based formulations as protective coating around fertilizer granules in order to slow down nutrient dissolution and improve fertilizer efficiency. The coating strategy relies on the formation of continuous polymer layer around the granules. This layer acts as diffusion barrier and controls the contact between water and the fertilizer core. The coated granules showed improved nutrient longevity with the release extended up to around 18 days compared to conventional uncoated fertilizer. From practical point of view, this approach is interesting because it combines bio-based materials, improved coating stability, and potential industrial scalability. Overall, this study showed that biopolymer coatings can be used as an efficient strategy to reduce rapid nutrient loss and support more sustainable fertilization. In addition to coating fertilizer granules, so we also developed a second strategy based on NPK-rich alginate-based slow-release beads. In this case, the nutrients are not only coated at the surface but directly incorporated inside the biopolymer matrix. The beads are formed through calcium cross-linking, leading to a dense egg box structure. These cross-linked architectures create tortuous diffusion pathways which slow down nutrient migration from the bead to the surrounding medium. The release behavior was evaluated in both water and soil. The results showed a sustained release over 35 to 56 days with profiles compliant with the European norm standard for slow release fertilizers. The release mechanism follows a non-frequent diffusion behavior, which means that nutrient release is governed by both diffusion and relaxation of the polymer network. Overall, this second fertilizer study, the biopolymer platform, can be used
J'ai
not
du mal
only
vous comprendre. Essayez de me demander à nouveau.
So overall, this second fertilizer study confirms that the biopolymer platform can be used not only as a coating, but also as a nutrient-loaded matrix for controlled and prolonged release. So the third application extends the same biopolymer engineering logic toward energy storage. In this ongoing work, bio-derived carbon materials are being explored as electrodes for supercapacitors. These carbon structures can be decorated with metal or metal oxide nanoparticles such as cobalt oxide, copper, zinc oxide, and nickel-based phases. So the design principle is based on three key parameters: porosity, active sits, and ion transport pathways. Porosity improves electrolyte accessibility, while the presence of metallic or metal oxide nanoparticles can introduce additional pseudo-capacitive contributions. At the same time, the carbon framework supports electronic conductivity and charge transport. The electrochemical response is evaluated through cyclic voltammetry, so the larger enclosed area of the CV curves indicates enhanced charge storage capability. Among the tested systems, the cobalt-based electrode shows the highest capacitive response with about 101 farads per gram at 10 millivolts per second and around 67 farads per gram at 400 millivolts per second, corresponding to nearly 66% capacitance retention. So the nickel-based electrode shows a lower but still interesting response with around 59 farads per gram at 10 millivolts per second and about 40 farads per gram at 400 millivolts per second. So overall, these results suggest that the same bio-based platform can be redirected from pollutant adsorption and nutrient release toward electrochemical energy storage. So this slide summarizes why our biopolymer platform can be transferred across different application fields. The first key element is the feedstock and chemistry, which starts from bio-based materials such as alginate-chitosan and biomass-derived precursors, which can be combined with mineral phases or nanoparticles. The second element is architecture engineered by tuning cross-linking density, grafting ratio, and mineral loading. We can control the internal structure of the material. So the third element is function control. This architecture directly influences surface charge, diffusion, pathways, and ion transport. So this is the core design rule of our work. So the same chemistry combined with tuned architecture can lead to a target function. This explains why the same platform can be adapted to water remediation, smart fertilization, and energy storage. In addition, the platform is multifunctional, valorizes local resources, and supports circular economy logic by linking pollutant removal, nutrient delivery, and clean energy storage. So to conclude, I would like to highlight four main messages. First, we established a biopolymer engineering platform based on alginate, chitosan, and related bio-based materials, designed as functional beads, coating, and carbon-derived structure. Second, this platform was validated for water treatment through the removal of heavy metals and dyes using alginate-chitosan hydroxyapatite beads. Third, the platform was extended to agriculture with slower release fertilizer coatings and NPK-loaded matrices that help improve nutrient use efficiency. Fourth, energy storage is now emerging as promising direction through bio-derived carbons decorated with metal or metal oxide nanoparticles for supercapacitor electrodes. Overall, this work proposes a circular biomaterial pathway from local resources to biocomposites, then to clean water, smart fertilization, and energy storage. And thank you very much for your attention.